Antibodies are the glycoproteins produced in response to the antigens by the cells of our immune system. They are also known as immunoglobulins. Immunoglobulins, the word itself suggests globulin proteins involved in the immune response. They are responsible for providing immunity to us against diseases. Immunity against diseases is provided by two mechanisms, innate or acquired. Innate immunity is the non-specific host defense mechanism of the body which fights against the infectious organisms invading our body. This is present by birth and is represented by the anatomy and physiology of our body. The physical barriers, the cellular barriers and the chemical barriers. The physical barriers like skin and mucous membrane, cellular barriers like phagocytic cells and natural killer cells, chemical barriers like cytokines and complements. Acquired immunity develops in the body after it encounters a foreign substance. Cells of immune system recognize the antigen and develop a specific killing mechanism for it. The initial response of the foreign antigen is slow but provides immunological memory. This memory enables enhanced and early response to subsequent exposure to the same antigen. Acquired immunity works through humoral immunity or antibody mediated immunity and cell mediated immunity. The humoral immunity or antibody mediated immunity is represented by the production of antibodies by B cells against antigens. Cell mediated immunity is the immunity provided by T cells, where the sensitized T cells, after recognizing the foreign antigen, attacks them and secretes lymphokines to initiate effector mechanisms. Three major categories of molecules involved in antigen recognition in the acquired immunity are antibodies, major histocompatibility complex and T cell receptors. Antibodies are important weapons of acquired immunity which possess greatest ability to discriminate against different antigens and bind them with the greatest strength. Occurrence in the body. Antibodies are present in our body in membrane bound forms on B cells, soluble form in blood, mucosal secretions and interstitial fluids in the tissues. The antibodies on B lymphocytes are present for the recognition of its corresponding invading antigen, as a result of which it gets activated. Soluble antibodies are present in serum and tissue fluids which protect us from invading microbes and foreign molecules. Antibodies in mucosal secretions and interstitial tissue fluids are meant for opsonizing the invading pathogens. Presence of antibodies in blood also work as diagnostic tools. If a person is suffering from any microbial infection, the antigens or antibodies can be detected in the serum of the patient. Serum can be recovered from blood by clotting the blood sample. Serum is a clear pale yellow liquid that can be collected from the whole blood after it is allowed to clot. Presence of an infection can be diagnosed by reacting this serum with suspected antigen. If the patient is suffering from the suspected disease, immune complex will be formed. If not, then there will be no immune complex formation. Bifunctional nature of antibodies. Two basic functions of antibodies are to recognize the antigens and removal of the immune complex. The antibodies work as identifiers for foreign molecules and tag them by binding to it. This immune complex now triggers effector mechanisms which destroy the microorganisms carrying those antigens or neutralizes the toxic foreign molecules. A series of processes which occur after binding the antigen and antibody in order to destroy the immune complex are the effector mechanisms. This involves the role of phagocytosis, complements and natural killer cells. To carry out these functions, the antibody molecule must possess two parts. First is the part which binds the antigen, and second is the part which initiates the effector mechanisms. Now is the elucidation of antibody structure. A series of experiments were performed by scientists to understand the structure of antibody molecule. Antibodies are globulin proteins involved in immune response. In 1939, Arne Sellis and Elwin Kaba experimentally proved that antibodies are present in the serum proteins fraction of the blood. They immunized rabbit with ovalbumin. They recovered this serum from rabbit after some time. The serum was divided into two aliquots. One portion was treated with the immunizing antigen, the ovalbumin, to allow the formation of immune complex. The remaining serum was recovered for analysis. Another aliquot, which was kept untreated, was used for electrophoretic analysis. Electrophoretic profile of untreated serum yielded 4 fractions corresponding to the serum albumin, alpha, beta and gamma globulin. Treated aliquot of serum contained serum albumin, alpha and beta globulin fractions, whereas the gamma globulin fraction was reduced significantly. This means that the gamma globulin fraction was used up in the agglutination reaction performed before electrophoresis with antigen ovalbumin. As the gamma globulin protein contributed to immune complex formation, we can say that the protein involved in the immune response are the globulin proteins, hence the name immunoglobulins. We now already know that the IgG, which plays the pivotal role in providing immunity against diseases, possess gamma type of heavy chain. Treatment of antibody molecule with enzyme for deciphering the structural details. To understand more details about the structure of antibody molecule, it was treated with papain. The immunoglobulin molecule was cleaved into 3 fragments. 2 of these were identical fragments of 45,000 Daltons, which retained the ability to bind corresponding antigen, thus named as FAB fragment, the fragment antigen binding. The digestion yielded one more fragment of 50,000 Daltons, which was found to crystallize when stored at low temperature. This was named as FC fragment, the fragment crystallizable. In another experiment, the immunoglobulin molecule was digested enzymatically with pepsin. This time the protein molecule yielded a fragment of 1 lakh Daltons, which bound to 2 antigen molecules. FC fragment could not be recovered as it was digested into many small peptides. Antibody molecule is composed of 4 polypeptide chains. The existence of 2 light and 2 heavy chains was ascertained by treatment of antibody molecule with beta mercaptoethanol. The disulfide bonds are cleaved by beta mercaptoethanol without any effect on peptide bonds. The digested protein was separated by chromatography. The results explained the presence of 2 larger polypeptide chains named as heavy chain of 50,000 Daltons each and 2 light chains of 25,000 Daltons each. Heavy and light chains both contribute to fragment antigen binding. To further decipher the details, antibodies were raised against the FAB and FC fragments of rabbit IgG in goat. The antibody against FAB fragment could bind with both heavy and light chains, whereas antibody against FC fragment bound to only the heavy chains. This made the picture very clear that both heavy and light chains contribute to make FAB fragments and the FC fragment is composed of only heavy chain. Isotypes of heavy and light chains. Next step was to decode the amino acid sequence of the 4 polypeptide chains. It was found that light chains exist in 2 types of sequences, which were named as kappa and lambda. These are the light chain isotypes. Both the light chains in the antibody were of similar type, either kappa or lambda. Heavy chain sequences were of 5 types, which were named as alpha, mu, gamma, delta and epsilon. These are the heavy chain isotypes. Variability in heavy and light chain sequences. All the antibody molecules have some basic structure, but they exhibit enormous variability in the antigen binding capacity. There are around 10 raised to power 6 to 10 raised to power 9 different types of antibody molecules in an individual. This variability lies in the antigen binding fragment. Amino acid sequencing of light chain revealed variability in antigen binding fragment. Antibody molecules with different antigenic specificities differ in N-terminal amino acid sequence. This variability was observed in 110 N-terminal amino acids out of 220 amino acids in light chain. Therefore, the N-terminal half of light chain is denoted as variable region. The C-terminal sequence of about 110 amino acids was one of the two types of kappa or lambda. Therefore, this portion is named as constant region. Antibody molecules initiate different effector mechanisms depending on their heavy chain isotype. The heavy chain of different antibody molecules were sequenced, and an interesting fact was revealed that the N-terminal 1/4 portion of heavy chain sequence was different in majority of the antibodies, but 3/4 C-terminal sequence was from one of the 5 types of chains. This explains the presence of nearly same length of variable region in heavy chains and 5 types of constant region sequences. The 5 types of heavy chains were denoted as IgG having gamma type of heavy chain, IgA possessing alpha type of heavy chain, IgM having mu type of heavy chain, IgD having delta, and IgE possessing epsilon type of heavy chain. These heavy chains initiate different effector mechanisms like IgG and IgM activates complements, IgA is transported to body secretions, IgE activates mast cells and basophils. Hypervariable regions on FAB fragments facilitate diverse antigen binding sites. The variable region of heavy and light chains exhibit extreme variability at 3 positions. These are known as hypervariable regions. These are the positions on antibody molecules which form the cleft for binding of the antigen. They are termed as paratope or complementarity determining region, that is CDR. Remaining amino acids in variable region form the framework region, which are responsible for maintaining the structure of CDR. Summing up the structure of antibodies, antibodies are Y-shaped glycoproteins which are made up of 4 polypeptide chains, 2 heavy chains and 2 light chains. The heavy chain is denoted as H and weighs around 50 kilodaltons, consisting of roughly 440 amino acid residues. The light chain is denoted as L and weighs around 25 kilodalton, consisting of 220 amino acid residues. 2 arms formed by the combination of heavy and light chains attached to the antigen and therefore known as fragment antigen binding and is denoted as FAB, whereas the arm formed by 2 heavy chains is known as the fragment crystallizable and is denoted as FC. Nearly half of the FAB fragment of the antibody possesses variable sequence of amino acids, thus it is known as variable region. The variability in the FAB fragment accounts for the property of specific antibodies binding to specific antigens. The structure of variable region is complementary to the structure of antigen to which it will bind. The variable region is composed of 2 parts, paratope or complementarity determining region, the site where antibody binds to the antigen, and next is the framework region, the sequence and structure of polypeptide chain which is responsible for maintaining the active site of paratope. The FAB fragment of immunoglobulin molecule lie towards the amino terminal end of the 2 heavy and 2 light polypeptide chains. Therefore, An antibody molecule possesses 2 Fab fragments, so it is said to be bivalent. Hinge region. The polypeptides of immunoglobulin molecule form a Y-shaped structure where the arms are formed by 2 light chains and half of the 2 heavy chains, and the tail is formed by remaining half portion of 2 heavy chains. The 2 arms can move freely and adjust according to the antigenic determinants, and the joining part of the 3 segments is known as the hinge region. Immunoglobulin domains. The heavy and the light chains are joined to each other by interchain disulfide bonds and many non-covalent interactions in the antigen binding fragment. The amino acids of heavy and light chains are folded in a specific pattern termed as immunoglobulin domain, which is stabilized by interchain and intrachain disulfide bonds. Approximately 110 N-terminal amino acids of light chain form a variable light domain VL. Similarly, variable region of heavy chain forms variable heavy VH domain. The constant region of light chain forms one constant light CL domain, whereas the constant region of heavy chain forms constant heavy 1, constant heavy 2 and constant heavy 3. Functions of antibodies. Antibody mediated effector functions include: first, the neutralization of microorganisms or toxic microbial products. Next, activation of complements. Another, the opsonization of antigens for enhanced phagocytosis. And lastly, the antibody dependent cell mediated cytotoxicity. Production of antibodies. A normal healthy individual of around 70 kg produces 3 grams of antibodies per day. Production of antibodies is a multi-step procedure including recognition, attachment, cross-linking, stimulation, storage as memory B cells, proliferation, differentiation, secretion of antibodies by plasma cells. First is the recognition and attachment. Antibodies are present as B cell receptors for antigens on the surface of B cells. These cells keep on circulating in the whole body, and whenever they come across their corresponding antigen, the invading antigens, they recognize the complementary structure and bind to them. Second is the cross-linking. The antibodies are bivalent in nature, therefore, they bind to 2 antigens at a time. The antigens are multivalent, so share the valencies of the antibodies. This leads to cross-linking of B cell membrane receptors. Next is the stimulation. The cross-linking of naive B cell receptors leads to its stimulation. Next is proliferation. As a result of stimulus, the B cell proliferates at a high rate. Then comes differentiation. Most of the clones produced by stimulated B cell differentiate into plasma cells. This differentiation is required to produce large number of soluble forms of antibodies. Secretion of antibodies by plasma cells. Plasma cells are specialized for high protein synthesis and secretion. Therefore, they synthesize huge number of antibodies and release them into the soluble form. Now, the memory cells. A portion of the cells are stored in the body as memory cells, which can respond faster when the antigen enters next time in the body. B cells encounter first time with its corresponding antigen, and production of antibodies and memory cells is the primary immune response. Class switching. Primary immune response constitutes majority of IgM type of antibodies and a very few IgG type. Subsequent antigen exposure, also known as secondary immune response, leads to fast antibody production consisting of more number of IgG than IgM type of antibodies. This shifting of class of antibodies from IgM to IgG is known as class switching. Antibody classes. The antibodies produced in our body are of 5 types and also known as the isotypes, depending on the type of heavy chains they possess. These heavy chains differ in the sequence of their amino acids. The kind of effector functions they perform also depend on the heavy chain structures. These antibody classes differ on the basis of size, charge, amino acid sequence, carbohydrate content. First, the immunoglobulin G. IgG is the predominant immunoglobulin in normal human serum and plays a major role in humoral immunity. It contains gamma type of heavy chain and accounts for 70 to 75% of total serum immunoglobulin. IgG exists in serum as monomeric bivalent form and weighs 1,50,000 Daltons. IgG is predominantly produced in the secondary immune response. This is the only isotype which can cross placenta. IgG proceeds for effector mechanisms by initiating the classical complement pathway. Next is the immunoglobulin M type. IgM is the first immunoglobulin isotype which appears in response to the foreign antigen. It is the leading isotype in primary immune response. It constitutes about 5 to 10% of serum immunoglobulin and contains mu type of heavy chain. Its monomeric forms weigh 1,80,000 Daltons. It is also found as membrane bound forms on B cells. It is present as pentameric form exhibiting star conformation. Next, we come to immunoglobulin A. IgA is secreted by plasma cells in dimeric form, where two monomeric Y-shaped structures are joined to each other by a J chain through C terminal of both the molecules. IgA constitutes 10 to 15% of total serum immunoglobulin. IgA is the most abundant immunoglobulin molecule in the whole body because it is present in huge amount in body secretions like breast milk, saliva, tears and mucus. Every day 5 to 15 grams of secretory IgA, that is SIgA, is secreted into mucus secretions. SIgA binds to invading bacterial viral antigens on mucus linings, which is the main entry site for most pathogens. IgE antibodies are involved in hypersensitivity reactions. They are present in serum in very low concentrations such as 0.3 micrograms per ml and contains epsilon type of heavy chain. Its molecular weight is 1,90,000 Daltons. It binds to Fc receptors present on mast cells and basophils and initiates its degranulation. IgE crosslinking on mast cells or basophils lead to the release of pharmacologically active mediators for antiparasitic defense. Next is immunoglobulin D. IgD is usually found as surface markers on B lymphocytes. Its molecular weight is 1,50,000 Daltons. It is present in low concentrations in serum such as 30 microgram per ml. It constitutes around 0.2% of the total serum immunoglobulin. No biological effector functions have been identified so far for IgD. Antigenic determinants of immunoglobulins. Immunoglobulin molecules can function as potent immunogens when injected in another animal species. The antigenic determinants or epitopes on immunoglobulin molecules fall into 3 major categories: isotypes, allotypes and idiotypes. First is isotype. Isotypic determinants are the constant regions present on the immunoglobulin molecules. The structure of isotypic determinants is determined by the structure of constant region of heavy and light chains of any given immunoglobulin. The constant region genes are specific for a given species and can be considered as foreign in another species. Next is the allotypic determinants. Allotypic determinants are the minute differences in the isotypic regions of different members of same species. This difference is due to the presence of different alleles of same isotype. Allotypic differences can be determined by injecting immunoglobulins from one member of a species into another. Next is idiotypic determinants. Idiotypic determinants are the variable region domains of any antibody molecule. The antigen binding fragment can be recognized as an epitope in a genetically identical member of species. Now, we come to monoclonal antibodies. An immunogen carries multiple epitopes and can stimulate multiple clones of B cells for antibody formation. The antibodies developed in natural condition inside any host are against various epitopes of an antigen. These types of antibodies produced are polyclonal antibodies. The heterogeneous mixture of antibodies produced inside the host effectively clears the antigen by initiating the effector mechanisms. Immunoglobulin's role of diagnosing the causative agent of infection by serological reactions can be better dealt by using monoclonal antibodies. Monoclonal antibodies are derived by stimulating a single clone of B cells specific for an epitope. This cannot be achieved naturally, but can be done artificially by hybridoma technology devised by Kohler and Milston. In this process, a myeloma B cell, which has the capacity of uncontrolled multiplication, is fused with a normal activated antibody producing B cell in the presence of polyethylene glycol. The resultant hybrid cell is immortal with the capacity to produce required antibody. This cell can be cultured and grown in large amount to produce huge number of monoclonal antibodies. Some of the applications of monoclonal antibodies are their use as in vitro diagnostic reagents in serological kits. They are also used for detecting pregnancy. They are also used for checking the presence of pathogenic microbes. Also for measuring blood levels of various drugs, matching histocompatibility antigens, detecting tumor antigens, and also used in tumor immunotherapy. So, now we are acquainted with the antibodies as important tools which have specialized polypeptide structure to inactivate targets by various effector mechanisms and protect us against infections.